Well, thanks for sticking around. It's only me and Amy that's in between you guys and lunch, so I'll try to be brief with my introduction. My name is Tess Weathers, and I'm a PhD student here at Mines. I work with Josh Sharp, also in the Geoenvironmental Microbiology Lab here. I am a civil engineer and hydrologist. I actually have a master's from UC Davis in hydrology, so did not know Scott at all because did not consider microbiology whatsoever. But now I'm getting thrown into it. I'm looking at the contaminants in firefighting foams, specifically perfluoroalkyl acids. You guys enlighten as chemists, and their impacts on bioremediation in the subsurface. I met Dr. Amy Buck yesterday and gave her that rundown. She asked me when I was graduating and what I was doing afterwards, and I said, I have no idea. And she gave me some very good advice. She said, just follow my gut, and that's what she did when she finished her PhD with the Norm's lab in 2005. She was one of the RNase P people. And after that, she decided to follow her gut and move to Scotland, where she did a postdoc developing nucleic acid biosensors. While she was there, she was awarded the Marie Curie Fellowship. So she started looking into microRNA and viral infections. They liked her so much there that they awarded her a couple grants, such as the Advanced Research Fellowship to start her own lab in the Center for Immunity, Infection, and Evolution at the University of Edinburgh, where she's studying RNA function and secretion in helminth models. And she wanted me to let you know if you guys want to try haggis or sample some good whiskey, feel free to visit her. Although I suggest don't stay as a house guest because it was actually her shower curtain that was used in that shower. So with that, Dr. Amy Buck.
Thanks very much for the introduction and the invite. It's more than an honor to get to speak in this context. And actually, the last time I spoke in front of Norm was at my PhD defense almost exactly 10 years ago. And the last slide that I showed was the acknowledgement slide. And as I gushed on and on about how wonderful Norm is and how much I loved the lab, I watched his face get redder and redder and then shrink in his seat. So I'm going to try not to do that too much today, also because I don't want to cry, and I will stick mostly to the science. I should mention one funny thing about the shower curtain. After I left the lab, I met someone, and I was saying I'd been in Norm Pace's lab, and oh no, I worked on RNase stuff. I said, Amy Buck, that was your shower curtain. I said, how did you know that? I said, you're acknowledged in the paper. So given the amount of press that came with that paper, that's pretty much what I'm known for from Norm Pace's lab. But anyway, so I'm going to talk more about what I've been doing recently. I'll start with a couple slides just of where I started in the Pace lab and sort of how that led different ways to what I worked on in Edinburgh. And hopefully I'll get into it. It's sort of come full circle, at least based on Norm's influence. So I was on the RNase P side of things, if I can go. I don't see it. So I was on the RNase P side of things, and Norm introduced this last night, and where really the enzymatic reaction that's studied is the processing tRNA, the maturation of tRNA. And I started my PhD in 1999, and it was intimidating because they had already done so much amazing work on the enzyme responsible for catalyzing that reaction, including discovering that the RNA had the catalytic activity, And then, as Norm also mentioned, developing really sophisticated and creative methods for probing the structure that were relevant, that's great, were relevant to other systems. Also, when I was there, Alexi in the lab solved a crystal structure, which proved to be really useful for my own work and was a wonderful achievement. So the PhD project was you got this big exciting RNA that's the enzyme. What's this little protein in bacteria? There's one protein. What's it doing? And really, what I learned from Norm is, well, you can't study something by just looking at one thing. And so my project actually was every RNA-seq bacterial protein we could make, studying it with multiple forms of the bacterial RNA. And this is not a model. This is a cartoon. If any of you know Norm, this is the cartoon that represents about 6 years of really hard work, basically showing that what we think the protein's doing, yes, it can stabilize the local active structure of the RNA. In some cases, depends how you look at it and which protein you're looking at, it can affect dimerization of the whole enzyme. That doesn't really occur in the presence of the substrate, and the protein can contribute some affinity, some energy to substrate recognition. And another piece of my work was really trying to then map where the protein binds on this enzyme. And this is just showing the B-type BST RNA, E. coli RNA, the red is where the protein binds. And again, this was using 3 different types of protein and doing the heterologous systems. And in a way, what I walked away from this is that, Norm already got it. He'd been studying the minimal conserved sequence for a long time, and others had contributed to that. And sure enough, the protein binds to that. And when Alexi solved the structure, we were actually able to map the protein into a nice little binding site on that structure. So that's all I'm going to really talk about. But the key lessons, I think the biggest, most important lesson is you got to see the big picture, and you can't get too lost in the details. And I think of this as with all those extra helices that aren't concerned. See past the bits and the bobs. I think Norm called them something else. See past the bits and the bobs. And as I say, don't get lost in the details. But some details are so important. For example, the salt concentrations that you use. I think that was required to discover that RNA had catalytic activity. And in my own assays, that Norm was asked. There's a reason he asked you what magnesium concentration you were working with. I'll never forget, after a month and a half of some gel assay, most of what I did was gels, not working, Norm coming out as he would, and sitting on it, what's the action? Which, it's just something I will always remember and treasure. What's the action? Oh, I can't get this, I'm not seeing this, I don't know what's going on. What about the NP40? And it was just, no, Martin, it's not the NP40. Now, of course I thought of that, It was the freaking NP40 that I wasted a month, and, one shot, and he knew what the issue was. So, the details matter. It's all about the data. Norm loves data, and I think sometimes you can get a little bit caught up in telling a story in a soundbite. It's good for getting funding, but I think he really helped balance that. Let the data needs to tell the story. It's all about the data. I think we all know this, so I'm not going to say the word. And don't say And I put this up because I think in addition to teaching us about being an experimentalist, being a good scientist, at least for me, I might not have appreciated it at the time. He also taught how to present your work, how to present it clearly, and how to try to write well. And I think that whole package, that's a high quality mentorship experience, and I don't think I can even match that. I try with my own students. They don't always appreciate interrupting them in the middle of the talk and saying, don't say on. The last thing, you gotta have a stomach of steel. I say this because that's what I remember. Norm used to say that whenever I get a rejection. He actually used to say it at all the RNA club meetings. things because you pretty much eat pizza left and right in grad school. But you gotta have a stomach to stay. So after 6 years in Norm's lab, where to go next? Norm mentions this yesterday. So this is a story from the 70s where Norm had found that 7S RNA is packaged into retroviruses. And this is the host 7S RNA, which turned out to be part of the signal recognition particle. And he was really interested in, well, why is this getting packaged? Is this involved in movement or trafficking? There's something there. This is interesting. Didn't know that he had been trying to sell this to anyone that comes to his lab since the 70s. But I actually thought that is really interesting. Why do retroviruses package 7S RNA into their units? And as I was introduced, I did kind of follow my gut thinking about a postdoc. And I'm like, you know what? I want to learn something about virology. There were reasons to go to Edinburgh with my partner at the time, and I thought, well, actually, I know nothing about virology, so I'll try to find a retrovirology lab. Couldn't do that. Ended up, at the time, microRNAs were gaining a lot of popularity, and they'd just been discovered that some viruses can make their own microRNAs. I ended up managing to get a postdoc in a lab where I could write a fellowship to study herpes viruses. I'll just touch briefly on that work, but then try to explain how that's led into my interest in parasitic worms and RNA secretion, how that led to finding vesicles, extracellular vesicles, where the question again is, why is RNA packaged into these vesicles? And it sort of basically landed right back 10 years later at the question. So, one introduction about microRNAs, I think everybody's heard of microRNAs by now. These are endogenous genes encoded in our genomes. They're generally Pol2 transcripts. Key feature of a microRNA is it's got a stem loop structure. Because of that, it's recognized by a protein enzyme and cofactors that catalyzes a cleavage reaction, trimming it to a shorter hairpin, passing it to another protein where it gets exported into the cytoplasm, further recognized by another enzyme, dicer, that cleaves it down into a 22 nucleotide duplex. And it's one strand of this duplex that gets incorporated into the RNA-induced silencing complex, and then the microRNA guides that protein complex to other nucleic acids, in this case generally messenger RNAs, through base pair partial complementarity. And these interactions lead to inhibition of translation. So if a microRNA is targeting a gene, that you get less protein produced. So what we know now, in 2015, there's on the order of 100 to 1,000 microRNAs per animal. Each microRNA can target multiple genes. So I kind of think about them like transcription factors. Yes, they're regulated networks. It's hard to imagine but they work in a different way, a pathway that's not regulated by microRNA at some level. And then where I got interested in them is they play roles in host pathogen interactions. So as I said, some viruses actually can make their own microRNAs that can interact with host genes. And that's an interesting kind of interaction. And what we've also found is that some viruses can actually inhibit them through non-microRNAs. What's new? What is new since about 2007, 2008 is the finding that microRNAs are also found outside of cells. And they're stabilized either through encapsulation in vesicles or bound to proteins. It's been proposed that their secretion and uptake is a form of cell-to-cell communication, and there's still relatively little known about RNA trafficking. So just to try to draw the connection between the viruses and the parasitic worms, I'll show one more slide about what my lab is really focused on. So here's the microRNA biogenesis pathway, where we have studied viruses and the fact, as I mentioned, that they encode their own microRNAs. They also can make nucleic acids to block mature microRNAs, and that's one of the fun stories that's come out. They can make their own RNAs that are complementary to a cellular microRNA, bind to that, and target it for degradation. What's interesting is we know very little about how you actually turn over a microRNA. So one side of my lab is trying to understand, okay, you use another nucleic acid, but then there's a whole turnover pathway that really is not understood. But where this links in with thinking about extracellular RNA is, again, if it's true that RNA is coming and going from cells, then you really can't think about steady state levels if you just ignore that. And it's also not clear if the degradation pathways might be somehow linked in any way or related to or influence export. So I did get genuinely interested in secretion based on this work, but also just because I had a chance to do something cool. And that was work with this parasitic nematode. This is actually a nematode that naturally infects mice. And so what I'm going to talk about is the finding that this nematode is secreting RNA and trying to study that. And there's also some interesting diagnostic implications of that work for infectious disease. So one more introductory slide. What do we know? What do we actually know about extracellular RNA? Like a lot of things, there's been a lot of interesting data out there for a long time, much of which is ignored. So it's been known that nucleic acids were in plasma since 1948. Then there were some interesting studies, I think even in the 60s and the 70s, showing that RNA could get secreted by cells. This field got really energized in 2007, 2008, because there were a couple seminal papers showing that microRNAs were in serum and plasma. And because microRNAs are decent biomarkers for some diseases, this opened up the idea that this would be a non-invasive biomarker. And there's been a lot of attention since 2008, I think, people just describing and sequencing microRNAs in every fluid you can think of. The NIH Common Fund committed $17 million to a big consortium in 2013, and I think this is the same fund that funded some of the microbiome work early on. And so people are interested, but there's still a question of, and there's diagnostic utility, I think, in this, but there's still a question of what is the purpose of this phenomenon. So now I'm just going to introduce the helminths. So when I moved in 2009 to the biology department, School for Immune Infection Evolution, there's a big focus on helminth research and Th2 type immunology there, and really good collaborators. And I was quite excited that we could actually start working with these models. So helminths infect a large portion of the world's population, and they're actually also a huge economic burden. They're a major global health burden, in some cases because they do cause really debilitating pathology such as river blindness, mostly in the developing world in sub-Saharan Africa, but also then because these are manipulating, changing the immune system, they're a big problem for co-infection. They have co-evolved with their hosts for very long periods. We know that they secrete immune modulatory molecules. They encode microRNAs because they're animals. So could these parasite-derived microRNAs and other RNAs interact with the host? And just to put this in context, my colleagues and the whole field has been studying what parasitic worms secrete for decades, but they focused on proteins. And the reason it's interesting to study what these secrete is not just to understand how they can establish chronic infections, but because they do actually have some beneficial properties. So there's a hygiene hypothesis that because these nematodes and parasitic helminths have evolved with us for so long, our immune system has adapted to them. And there is a higher incidence of autoimmunity in developed countries, and some people think this could be because we've gotten rid of all the worms. So people have been isolating some of the products that they secrete, and some of these do have beneficial properties in allergy or autoimmunity. Anyway, so the question is, great, we know they secrete proteins, and those are interesting proteins, they have immune modulatory properties. Have they figured out how to specifically and effectively get RNA into cells? So the model that I mentioned is H. polygyrus. This is a gastrointestinal nematode that establishes a chronic infection in mice. It's in the same clade as C. elegans. The life cycle in the lab, at least, starts with the infected larvae. You introduce these to mice by gavage. They undergo a series of developmental molts, I guess, into the adults that you can detect in the small intestine after about 8 days. And then these wrap around the villi of the small intestine, and they can persist there for very long periods. And they actually, you have males and females, and this is worm pornography. I caught them in the act of mating in that lovely environment, and then the eggs get expelled in the feces. And so that completes the life cycle, and the larvae develop from the eggs, and normally the wild mice ingest these. So the way you study the secretion, one way you study the secretion product is to take these directly out of the mouse and examine what they're pumping out. And the beauty is this, as I said, this has been done for a long time by others. They've dealt with the artifacts and the worms dying, figuring out the conditions to try to get this right. So where I started really was just taking this secretion product and then concentrating, extracting RNA. And this is just a PCP labeled RNA, so polyacrylamide gel, with the eggs, the larvae, the adults, and the secreted material. And you can see this band, this smear around here is where you can see microRNAs, and you see a population. You also see a couple other distinct bands in the secretion product, which we are characterizing. And when you make standard libraries, so we're only really looking at stuff with a monophosphate here, 5' monophosphate. You see that in the adult library, yes, majority of your RNA is microRNAs. You have some uncharacterized, you have high RNAs that are in the germline, a little bit of ribosomal small RNAs. And in the secretion product, again, the majority of what we see in that library are microRNAs. And so the question is, well, which microRNAs are being secreted? And this is just showing a heat map where white is highly abundant and black is not abundant. Where you look at the most expressed microRNAs across different life stages, in eggs, larvae, adult, and the secretion product, and you can see, as you expect, there's some that really switch on or switch off during development. And that's how we know microRNAs work. They play a lot of roles in development. So one example is the MER35 family, which is enriched in eggs, and that's involved in embryogenesis. But then, interestingly, you see another microRNA that's turned on in the adults, LET7. Okay, that makes sense. It was the second microRNA ever discovered, plays a role in developmental timing, but it's also secreted. And so you think, why would a microRNA that's involved in worm development get secreted? There could be a number of answers, but one thing that's intriguing is if you look at that same microRNA is conserved in humans, and it actually has a lot of interesting functions. One of its targets is IL-6. So it could be that it might be doing one thing in the nematode, but if it does get secreted, if it does get into those cells, it could be doing something else. So in order to have to be present in order to do anything, these things, microRNAs have to be stabilized somehow. So this is where from looking at the literature and this new exciting data on the fact that vesicles carry RNA, I basically just ultracentrifuged my secretion product, extracted RNA, did a PCR against 4 of the microRNAs, and you see that compared to the input, the majority of your fraction is pelleting upon ultracentrifugation. And if you look at that pellet by TEM, you see these little vesicle type things. So the model is that the nematode is secreting RNAs, they're packaged in vesicles, and that's what stabilizes them. And just to sort of group that a little bit further, if you treat the vesicle fraction with RNase, you're largely protected. The microRNAs are protected. If you treat with Triton to break the vesicle and then RNase, you see these are degraded. Another interesting observation is microRNAs are not the only RNA that's secreted. One other thing that we picked up in these libraries was actually these yRNAs. And those are yRNAs are conserved. They're present in human, present in nematodes. But there's not that much that's understood about them. They've been shown to be involved in DNA replication. They bind to rogue proteins. We have no idea why they're in these vesicles. What's interesting is it's the full-length form that we see in the vesicles. When you break the vesicle, the full-length form gets converted into a smaller fragment. And we actually see a lot of this fragment in the non-vesicle secretion. So what this tells me is, these vesicles really are protecting the RNAs that are in them from natural degradation, the nucleases that could be in the outside of the vesicle. But also it may be that our methods, we're breaking 90% of our vesicles. So we're actually looking at, why, yes, we're protected here, but then clearly there's a lot of this other stuff outside of the vesicle that's got to be coming from somewhere. So I won't go too much more into that right now. So it was exciting because no one ever characterized vesicles being secreted by this type, by a nematode. And so we also want to do proteomics to see, to learn more about what these vesicles were. And what I'm showing here is a heat map of mass spec data, so 3 replicates of the vesicles from the nematodes and 3 replicates of the rest of the secretion product that doesn't have the vesicles. And you can see that there's absolutely enrichment, so this is the p-value and this is the vesicle to supernatant ratio. These are your vesicle proteins, these are your non-vesicle proteins, and then these are the ones that are both. And so what we see specifically in the vesicle proteins, we see proteins that are known to be in, I'm not going to talk about that, known to be in mammalian vesicles, so tetraspanins, heat shock proteins, Alex, Rab proteins. We also see specifically a lot of proteins that are known to come from the intestine. And if you actually look, this is the nematode intestine. If you look, you can see vesicle-like stuff. It's hard to know this isn't food for viruses, but we're looking now to better track where these vesicles are coming from using antibodies. So you see exosome proteins, that's the type of extracellular vesicle we think these are. You see intestinal proteins, and then what got me really, really excited is you see a secreted argonaute protein. Argonaute is what binds to these microRNAs. And what do we know about argonauts? So some argonauts are really, really highly conserved and ancestral. And those are the ones that bind microRNAs, which are in most animals. Nematodes have evolved a whole clade of argonaute proteins. And there's a lot of functional diversity in RNAi pathways in nematodes. So argonaute proteins and RNA are involved in epigenetic inheritance. They're involved in environmental sensing. So there's a lot of new biology in nematodes with this pathway. And what's exciting is understanding whether or not the nematode argonaute, we're looking, is that what's bound to these RNAs in the vesicles? And does that actually function in a mammalian system? And if we're trying to think about potential RNA-mediated cross-species communication, I think the proteins that are bound to the RNA are really important. So, but if we even want to think about that, we need to know that the vesicles and the RNA is actually getting into a host cell. So if you just label the vesicles with the lipophilic dye and incubate these with mouse intestinal epithelial cells, you can see uptake to the cytoplasm. It requires ambient temperature. You could do PCR on the host cells following incubation and get a signal for your parasite sequences. You can detect actually the yRNA by northern blot in cells after you've incubated them with exosomes. And actually what's worth mentioning is, in addition to microRNAs, we're characterizing them. So these vesicles get taken up by host cells. You can detect the RNA in the host cell. What are they doing? So we did one microarray experiment, just looking at the host genes that change when you put the nematode vesicles onto them under conditions where we believe we're getting decent uptake. And this is, again, is a heat map where you look at things that are going up and things that are going down. And I'm just going to circle a couple genes, DUSP1 and IL-33 receptor, which are genes that could be involved in this infection. So DUS1 is involved in the MAP kinase pathway, and it regulates IL-10, which is an important cytokine for this infection. IL-33 receptor is a receptor for IL-33, that's a key alarmin. It also sort of triggers the Th2 type response. It makes sense that the worms would want to inhibit that. So this is where you get a little bit conflicted, because after doing mass spec and RNA sequencing, I've come to appreciate these are really complex little entities. And the story I wanted to tell was that microRNAs are functional. Secreted microRNAs are functional. But secreted microRNAs are in a really complex little package, and it's hard to know who's the functional unit. So all I'm going to show, all I'm comfortable showing right now, proof of concept, that if you take the DUS1 3' UTR and you say, could the nematode microRNAs be targeting it, and you look at the conservation of that UTR and where any of the nematode microRNA binding sites are, yeah, there's a few sites close together. IL-33 receptor 3' UTR is not conserved at all. We have no evidence that the microRNAs are regulating it. But what we found, if you just take that DUS1 UTR sequence, you fuse it to a luciferase reporter, and you transfect the nematode microRNAs, you can see a reduction. So LET7 is one of the microRNAs, MER200, MER425, and if you put all three of them together, you get a stronger suppression. So one model is, as I mentioned, a lot of the microRNAs that are secreted by this nematode have functional homologs to host microRNAs. So there are already existing target networks in the host cell and conserved sites. And yes, under some conditions, if you put a cocktail of those nematode microRNAs into the cell, they can regulate these genes. Where I'm intrigued and a little frustrated at the same time is this is just proof of concept, and we really can't move forward until we do more biochemistry. And an example of that is if these work with the nematode Argonaut protein, we should be studying that complex instead of just chucking in RNA and assuming they integrate into the host pathway. So, with that, we're just going to go to one other question, is detection of secreted RNAs in vivo. So, this was all in vitro, which is great. But you want to know that these are actually being secreted in vivo. Because it's a gastrointestinal nematode, we can't readily detect them in serum or plasma, which is what we often sequence from. But what we did do was take another mouse infection model, Solidomycosis sigmodontous, which resides in the pleural cavity. And when we sequence the serum from mice infected with Lidomycosis, we see microRNAs, nematode-derived microRNAs, and we see several that are homologs to the H. poly microRNAs that are secreted in exosomes. So I think this is a little bit of, this suggests that, first of all, this is a conserved phenomenon. All the nematodes we've looked at secrete RNAs. Some of them are common, whereas you have some that are different in different infection models. And we're actually now doing this with human parasites because there's some interest in developing this as a diagnostic. So initial conclusions, RNAs are secreted by nematode parasites. Many of these are packaged into exosomes that can functionally suppress host genes. And I think there's a lot of implications, at least, for cross-species communication in the field, if we think it's a field of RNA trafficking, but it really needs developing. And then there's some interesting therapeutic and diagnostic applications. And what I'm just going to end with is full circle. Norm wanted me to study why 7S is in retroviruses. And what I've learned by having to learn about these extracellular vesicles is actually it's really hard in some preps to distinguish viruses from the vesicles. There's a lot of really dirty preps out there where people haven't appreciated that a lot of the vesicles aren't the virus. You have both. You have stuff coming from the host cell, either through this multivesicular body pathway. So these are the exosomes. They come from an endocytic pathway. Or you also have lots that's actually budding from the plasma membrane. And there's a hypothesis, the Trojan exosome hypothesis, that HIV really uses aspects of this pathway, this host mechanism, in its life cycle and also for immune evasion. So why do viruses package 7 siRNA into their virions? Well, they may be using a conserved pathway for transport of material between cells. Cellular RNAs, proteins, and lipids are packaged into vesicles normally. And why the RNAs are there, we still don't know. They could be involved in packaging, export, import, gene silencing. And is extracellular RNA trafficking a legitimate concept? And so, I do think it's kind of funny that because I feel passionately now about this, why didn't I start with retroviruses? Why did I have to go through herpes viruses? It's been an interesting path. But we're organizing a meeting actually. Extracellular vesicle-associated RNA is their purpose, and that's in a few months. So I'm back to where I should have started. And I just want to acknowledge without crying, can't say enough about Norm. The lab was an amazing place to be. I was there between 22 and 29, the best years of your life. I thought for some reason I wanted to spend most of my weekends living in Boulder in the lab, but it was truly an amazing time to be there. Thanking my group in Edinburgh, our funding, and some wonderful collaborators in Edinburgh. And thank you for your attention.
Generic questions. Are there certain cell types in the world that express exosomes and not?
Yeah, no, it's a good question.
And then the other one, which is probably more interesting. How are they selected for different kinds of RNAs within the cell? How do they?
So the first question is, I don't really know. I mean, we think these must be coming from their intestinal origin. In organisms, I think vesicles, this is an organism, vesicles are involved in cell-to-cell communication. So, yes, probably they're produced by other cells.
Specific microRNAs can be chosen for.
Yes, so that's the key question, because the null hypothesis is just garbage, right? Cells are getting rid of stuff. And I forgot to mention that if you put the exosomes do have some interesting immune suppressive properties, so we don't think they're garbage, but where is their specificity and energy devoted to packaging? And what it's hard in this system because we can't culture and isolate the cells from which they're derived, and that's what you have to look at to look at specificity. But what people have seen in mammalian systems, and we're trying to sort of be able to do this as well, is that you do have a selective set, for example, of microRNAs that are packaged. And how is that happening? Because it's a 22 nucleotide sequence. How do you get specificity? And some people have proposed, there's a motif and there's some RNA binding proteins that are shuttling. I think there's enough evidence to show that there is selective export, but the mechanisms are still not totally clear.
So if there's selective export, and when the nematodes are grown not in the mouse intestine, you would think they would secrete different microRNAs, perhaps no microRNAs.
Yeah. No, it's something we want to look at, and that's why I think we're going to switch a bit more to the filarial model where we can see. I mean, we know in vivo that the RNA is getting, so we can detect the RNA in vivo secreted in the human fluids, and some of the homologs from that model are the same as what we see in vitro with H. coli. But the answer is absolutely. You would assume this should be dynamically regulated, and the conditions matter, and you would see differences. So we're looking at that.
So I know about here produce vesicles and they package plasmas and stuff like that. It's been soft seeds. Do you have any way to look for microRNAs? Is that now I'm thinking what it is?
Yeah, I mean, it seems like vesicles are everywhere, and they are produced by archaea, bacteria, eukaryotes. That you don't have all the same type of RNA, and I don't think, do archaea have microRNAs? I don't think so. They have some of the proteins. I think they don't have the classical microsomes, but absolutely, I'm sure that RNA is secreted, and it may be relevant actually to do more comparative analysis on what is common in all these vesicles. But there is diversity in the vesicles. I mean, some are, and I think the things that are coming from the endocytic pathway may have a slightly different package than what's coming from another route. But yeah, they're everywhere. And I think from kind of curious people who've been studying microbiome, right, they're everywhere. And the exosome field, the vesicle field feels like that right now. And why are they everywhere? Why are these things, what's the niche in biology where you need a vesicle, a vesicle type communication? But it's a good question, yeah.
So I have a semi-retired colleague across the hall, some of you may know, Mike Yaris, who has been obsessed with the idea that RNAs bind specifically to membranes.
Yeah.
And that there's membrane-specific association between RNA. I wonder if that's a way you can achieve specificity of transport,
Yeah.
especially since argonaute is going with it, maybe a bigger RNA being processed that way.
Yeah, absolutely. And I think actually before I figured out that those hairpin sequences, that they were YRNAs, because they're really abundant in the vesicles. I actually found one of Mike's papers with Anastasia, characterizing the type of RNAs that found in membranes. This is it, because the hairpins. Turns out there's a lot of hairpins everywhere. But I think it's very relevant for understanding where these RNAs are localized in the cell and how that could affect export and specificity.
Thank you, Amy.